Okay, continuing on with Alzheimer's disease, what we're looking at here is if these are the healthy cells over here, if you look at these neurons, you know, they have this really nice healthy body in here compared to this Alzheimer's. Look at that cell body. It's all crooked and kind of messed up, and it's not really that healthy, right? Well, you look at that, but what you're really seeing, the part that really stands out, is all of these things in here are the neurons are all tangled up. They're going every which way, and you got these big old bunches of them everywhere. Okay, so that's what you're seeing with these plaques, they're called, or beta amyloid plaques found between these cells, and then you get these tangled, twisted strands of the proteins all messed up, and they're in the same place. So that's, again, beta amyloid plaques and neurofibrillary tangles, neurofibrillary tangles, neurofib, F-I-B-U-L-A-R-Y, tangles, and that's what you have. If you look at right here, it says, you know, oops, I just totally messed it up, but it says tau proteins. Okay, so tangled clumps of tau proteins is what we're talking about when you compare that little drawing with right up here. Okay, you'll hear that because they technically are tau proteins, or you hear them called neurofibrillary tangles, or tangled or twisted strands of proteins, or something like that. And basically what's happening here is, if you look at this microtubule over here, and it's healthy, but then all these little subunits of that, they start falling apart in little pieces, and then all you get after that is just a big old mess of that protein. Okay, so again, neurofibrillary tangles, beta amyloid plaques, highly pathognomonic of Alzheimer's disease. Okay, now if you look at this big old brain here, what you're seeing is, if this side is a healthy brain, this side is, again, very advanced Alzheimer's. But if you can see where that cortex is not only, it's shrinking, but everything inside of it is shrinking also. Okay, that area actually where I put the arrows is part of your hippocampus or hippocampus, and your ventricles get really big. So these big spaces in here where your cerebrospinal fluid flows and down through there, the lateral and the third ventricles and so forth, but they're not supposed to be that big. Okay, they're supposed to be smaller. The fluid flows through there into the third ventricle and so forth and down, and it's not supposed to be that big. Okay, so the main thing here is your cortex is all shrunken up, and you get the big ventricles inside there, and your hippocampus, which is a huge part of memory and things like that, which is what we're talking about, right? The hippocampus, the hippo never forgets. Well, that's how I remember that it has to do with memory, and of course Alzheimer's is a major memory problem, at least in the first stages, before it turns worse. Okay? Sorry, Huntington's disease. Moving on, Huntington's is pretty unique. You don't really see a lot of that. Now, it says right here, and I'm going to kind of do this a little bit out of order. It says right here that you tend to see the onset, as in when it starts, around 30 to 50 years. But because this is a genetic thing, a genetic disease, patients are born with it. Okay, so it's autosomal dominant, which means that it will be expressed. Okay, you don't have to have two of them, and it's on chromosome 4. Okay, so those are the genetic aspects of it. But you're born with it, or not you, but Huntington's disease patients are born with it, but it doesn't kick in until they're 30 to 50 years old. So this is one of those things where if a parent has it, then the child can get genetic tested, and it's not going to change the fact that it's autosomal dominant, or that it is probably going to kick in their 30s to 50s, but it does let them know what to prepare for. Okay, now obviously something like that you don't typically see being passed on purpose, but by the time somebody is 30 to 50 years old, and they get it, then they've already have reproduced, and now, because people are living longer, it stands out more that it might run in the family, but it might not have before when people weren't living that long anyway. Okay? Anyway, so what's happening, again, it's just like as far as, Alzheimer's goes, it's still progressive degeneration, and we are talking about nerve cells. Okay? Now GABA, if you remember what GABA is that if this is your neuron, and your electrical impulse comes down the neuron, and is supposed to, take these little whatever they happen to be, your neurotransmitter, and send it across the cleft into the, whatever it happens to go to, muscle tissue, say, for example, in that cell. Well, GABA is inhibitory, okay? So what GABA will do is it'll go ahead and it will cut that off, okay? Not literally like that, but it'll cut it so that the impulse doesn't continue on and nothing makes it that far. Okay, so GABA is really good, and it's useful in like opioid drugs for pain, things like that. It'll shut off that pain impulse. Okay, obviously it's not good when you are trying to move your muscles, and that's one of the problems with Huntington's disease is you lose that GABA from inside of your brain in the basal ganglia area, again, center of the brain. And without that ability to stop things, you can't stop your muscles from twitching either. Okay, so GABA is not just used in pain avoidance, but it's also used for many processes that are normal within the body. Okay, what they call it is chorea. In the PowerPoint that you should have available to you, you should be able to click that and it'll take you to a little video of it. You really should. I'm not going to waste the time on this video making links to other videos, but you should definitely go to the PowerPoint and look at these videos, and when you do, you'll see the differences between Parkinson's and Huntington's, and I'm not sure if there's Alzheimer's video or not. I don't think there was. But anyway, the Huntington's is, of course, irregular, and it's spazzing. Okay, so, spaz is not flaccid. It's tightening. Okay, so you're getting these involuntary jerkings and movements that come with it. Okay, it can be the face, the limbs, or the whole body. And hyperkinesia just means too much movement. Okay, so you see literally flailing around where the arm just shoots out into nowhere. Sometimes you hear the phrase Saint Vitus dance. Again, another buzzword, I guess, for that. But the Huntington's chorea, a lot of times they'll simply call it Huntington's chorea instead of Huntington's disease because of the association with that hyperkinesia, with that movement all the time. And this is typically more movement than you would see even in Parkinson's disease. Okay? All right. As far as dementia goes, that's why it is in this unit. In dementia, you start losing your executive attention deficits first. Okay, so a lot of times Huntington's, you'll be talking about higher brain functions, maybe balancing your checkbook, making executive kind of decisions, that sort of thing. All right? And Brady, you haven't heard this word before, phrenia, probably, and that's just your thoughts. Okay, so you have a slowed down thought process, and it also affects your mood. People get depressed. Not exactly sure if that's because of the deterioration of the basal ganglia or not, but it certainly would make anybody depressed because even though they are slowed thought processes and they can't think as well as they used to, it's not like they are completely out of it. They understand frequently what exactly is going on with them, and that would depress anybody, I would think, but hard to say exactly what the biological or psychological balance is between that. Okay, all right, so not only do you have the dementia and the movement disorders, but you also have, of course, problems with speech. Okay, so as those functions, as the basal ganglia gets deteriorated, speech is also an issue because, of course, with anything, you still need the nerve movement going down that axon. And if GABA is not inhibiting anything. It goes too fast, it messes everything up. It also throws off your balance for pretty much the same reason. Too many impulses, right? Too many impulses not being regulated, I guess, is a good way to look at that, because GABA would regulate things, and as in slow it down and stop it if need be, but it can't do that, obviously, if you don't have your GABA there in the first place. Okay? Always good to look at CTs and MRI. This is one of those things that you would use to not only diagnose it, but rule out other things. So the caudate nuclei atrophy, basically it's not important that you memorize at this stage in your lives what the caudate nucleus is, but again, it's part of that basal ganglia, and it atrophies. Okay, so it starts shrinking down, and you'll see that on a CT or MRI, and of course your genetic testing that you're looking for that chromosome 4, right? Okay, here is the picture, I guess, if you really cared about the caudate nucleus down there, which is right in the center of the basal ganglia portion of the brain. All right, I don't think you have to know about all this at this moment, you know, the globus pallidus and the putamen and that sort of thing, the thalamus. Anyway, this is all in that area, the part of the brain that's getting affected. As you see, the important part is right in smack in the center of the head. Okay, so it is very, very, very difficult to really do much with, even if you could do anything about it. Okay, 5 in 100,000 patients is how many it affects. Again, if it's not kicking in until maybe 30 to 50 years old, and the patient lives with this disease for 10 to 25 years, well, you're talking, up to 75 years old you could be if you started at 50 years old. So again, in some cases a pretty long life. On the other hand, if it was an early onset and it only made 10 years, then you're talking death at 40 years old, which is obviously not very good. Okay. Pneumonia, cardiovascular disease were the most common causes of death with Huntington's. Again, I think that has more to do with the fact that you're in the hospital because of the dementia, and a lot of people die from pneumonia in hospitals despite how hard we try to keep it out of there. Okay, again, the mean age was more of a mathematical thing. 51, 57, that's not as important as, fairly young. These people are typically dying. Typically, again, you can make it a lot longer, or you can make it even much shorter. All right, Huntington's is one of those. It does have different levels of penetrance, meaning that you can have very severe cases and you can have not so severe cases. It really kind of just depends on how bad it is. Okay. Now the CAG, what is that? Cytosine, adenine, and guanine, the amino acids CAG. What's happening here is Huntington's gene has over 28 repeats. Okay, so when you start an amino acid, then say it's A, U, G, and then Huntington's would be like CAG, 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 and we can, in our genetic code, we can splice things out, to a certain extent, with your exons and everything, but when you start getting that many of them, it is a little bit much for even our internal mechanisms to completely get rid of. So that's why you would see varying degrees of Huntington's, and why so many of those repeats causes all these problems. All right, now if you look at here a little more detail here, if you have less than 28 of those CAG repeats, then you're talking normal. Okay, we can catch that, as the internal mechanisms can go ahead and catch it, and you're not going to get Huntington's disease. It's not going to manifest. Okay, 29 to 34 repeats of the CAG go in there. If you do that 29 to 34 times, then the person is still not going to get Huntington's, but the next generation is a lot more at risk, because you remember that, as you're doing that, what's that T? Anyway, you're transcribing that along, and eventually, if you're passing on a whole bunch of CAG repeats to start with to the next generation, then they are a lot higher at risk for getting Huntington's, because they could also, assuming they have the same chromosome 4 problem, they could also add on to that, and now you're talking a lot more. Okay, now when you're talking 35 to 39, some people will get Huntington's, but not everybody. Okay, again, we do have mechanisms to cut things out and to get rid of them. So even at that stage, but now when you're talking over 40, okay, that's a problem, because now you're gonna get Huntington's disease, and that's the typical one that we're talking about. Okay, when you're talking about the ones that 35 to 39 repeats or something like that, maybe that's a less severe case. But when you start talking over 40, obviously the more over 40 you get, the more severe the case will be. Okay, so again, in summary on that part of it, it's not that the repeats themselves can't be dealt with our body, it's just that how many of those repeats can stress out even our own factors that we use to cut out unnecessary repeats and diseased genes. Okay. This is a little article way back from 1965, okay, which is a long time ago. They used to, they were studying fingerprint patterns, and to try to find out, about if somebody was at risk of having Huntington's disease. Again, it's way back in 1965. So they took these fingerprint patterns of these patients with Huntington's. Again, remember I said they call it chorea sometimes. And they took 50 with Parkinson's also, and they compared them with Scotland Yard, because of course, like the FBI of England, they have all these fingerprints and everything. They saw more of the whorl pattern. Again, the whorl pattern is another thing that you kind of hear with Huntington's disease that has a bit of a buzzword also. And but they say you can't actually diagnose or even say prognostic how bad it's going to be when you have that whorl pattern in your finger. So this is whorl pattern, is more of Wilson's disease as far as the more pathognomonic, but again, you can see it sometimes in Parkinson's and sometimes in Huntington's. I'm not actually sure, now that I think about it, what the point of this little thing was, but Wilson's disease is a whorl pattern one, and you might find it in other diseases, but they're all talking about some kind of dementia. Okay. Urinary tract infection. Moving on, changing subjects there, is again, it's a common cause of confusion in the elderly. Okay. Now, the thing is say somebody has a cystitis, or a bladder infection. Usually talking about E. coli. Okay, pyelonephritis. Again, you get into the nephrons and inside the kidney itself, and these are just urinary tract infections, right? And because of these urinary tract infections, they can get pretty bad. Okay, and when they do get really bad, it can actually cause a delirium, really, or confusion. Delirum, sure. A delirium or a confused state. Okay, well, again, just because they're elderly, you don't automatically say that you think that they have Alzheimer's or something like that. Send them on their way. Sorry, there's nothing we can do from you, especially because, obviously this kind of delirium from a urinary tract infection is very curable, right? So, increased females, again, if you're talking about they have a shorter urethra, therefore these E. coli and other bacteria can get in there faster and get back up to the kidney. Because of that shorter urethra, diabetes patients have an increased risk of any urinary tract infections just because of the massive amounts of sugar. Catheters, of course, although they go way out of their way not to introduce pathogens through catheters when they're putting a catheter in somebody at the hospital, occasionally it still does happen. Okay, poor hygiene, of course, this is more of a female kind of thing where they're wiping the wrong way and bringing pathogens into their urinary tract and causing infection that way. Dehydration, again, water will flush things out. Lack of water makes it harder to flush out, and always, of course, a compromised immune system is a risk for pretty much everything. Okay, so painful dysuria, so painful urination, frequency as in you have to go a lot. When you have to go, you really have to go now. It's urgent. Okay, any kind of fever should definitely be a big red flag, because fever has more to do. You should start thinking more infection than if somebody comes in seeming like they have Alzheimer's, but all of a sudden they have a fever, right? And that's where you go, okay, oh my goodness, this person has a fever. Let's check the flanks, the small of the back there on the sides, and see if the kidneys hurt, especially if the person's nausea or has nausea and vomiting, something like that. Yeah, okay. Blood in the urine, of course, from the infection and leukocytosis, it's going to be the white blood cells are going to be increased because there is an infection. Okay, so again, urine analysis, urine culture are the testing for this. So urinary tract infections, I think they're pretty straightforward. The problem is making sure that you just don't automatically assume that just because they are old and their memory, or they seemed out of sorts with their consciousness, that you don't automatically just say, oh man, they got Alzheimer's or something like that, and let them go home. Okay. Hypoglycemia, I don't know that this should be much of a surprise to anyone, right? Hypoglycemia is not enough blood glucose, okay, or glucose in the blood. Why would you get that? Well, duh, not eating. You could do that. If you don't have enough sugar, then insulin shock, of course, is a unfortunate, fairly common one where somebody is diabetic and they're taking insulin and they just they take too much of it and drop that blood sugar way too low. And alcoholism is another reason, of course, because the chemical reactions that you would have with the sugar. Hepatitis goes a little bit with the whole idea of alcoholism, because again, they're both related to the liver, and when the liver is either inflamed or trying too hard to get rid of toxins, then you could literally burn too much sugar out of the system. Okay, and then of course there's like an endocrine tumor where you are actually making something, I don't know what, that you're using too much sugar because you're overproducing something, say insulin, for example, and making too much insulin that'll drop your blood sugar level. Okay, so what are the signs and symptoms of not having enough blood sugar? Well, you get hungry. That makes sense, because that's what hunger is not knowing you have enough blood sugar. Going to send the signals for that. You're going to get cold and clammy. Okay, clammy is kind of like moist or wet, and cold is cold because, again, not enough blood sugar. You're dropping your energy levels, just like somebody with too much blood sugar gets really hot and sweaty. Okay, headache will be caused by that. Anxiety, you're going to feel very anxious if you don't have enough sugar in your blood, just like you would if you had way too much. And of course, arrhythmia, same thing, right? Too much sugar or too little sugar is going to cause heart palpitations. or palpitations, I guess. Palpitations is just feeling it. Anyway, testing. Okay, duh. If you think it's hypoglycemia, test your blood glucose, and you do it fasting and 2-hour postprandial, they say, or after eating. Prandial is eating. Okay, so again, 2 hours after eating, you test your blood sugar, and also fasting blood sugar, and that'll tell you if you have the propensity to have your blood sugar drop too fast. Okay, some people just really do drop blood sugar really fast, and you would go ahead and do those tests and find out if in fact that's one of those people. All right, and of course when somebody does have really common hypoglycemia, they get a little medical alert bracelet, so you can see that on there. So if somebody starts shaking and going through all of these signs and symptoms, I guess I didn't put shaking on here, but of course shaking is really big. But if you start seeing something like that, then definitely start looking at their blood sugar and look for an ID bracelet if they have one. Okay, liver failure. Oh, I am way over time. I guess I got to break this one up to another slide.